This figure is just showing the absolute basics of resistance evolution. You have a population, it's sensitive to whatever it is that the population is going to be exposed to. For convenience, we'll just call it an antibiotic. Within this population, you may or may not have individuals that in fact are resistant to that antibiotic. If they are resistant, that resistance could be a consequence of mutation. It also could be a consequence of horizontal gene transfer. But nevertheless, the population predominantly consists of sensitive individuals, and that is particularly so if there is a cost that's associated with carriage of whatever the allele is or alleles that are associated with resistance. You add to the population the antibiotic or whatever it is the population otherwise is sensitive to. The population crashes as a consequence, and coinciding with that crash is an increase in the frequency of whatever individuals are in that population that in fact are resistant to, in this case, the antibiotic. That frequency will increase until, in fact, given sufficient exposure to the antibiotic, the resistant allele will reach fixation or something close to fixation. If this were a community, instead you would have a community that consists of both sensitive and resistant individuals. We'll say here that in the absence of the antibiotic, most of the individuals in fact are sensitive. Again, this would be assuming that there's some cost probably to resistance to the antibiotic. And then when the antibiotic is applied, the community declines in terms of numbers of sensitive individuals, species of sensitive individuals, and you get an increase in the frequency or numbers of the individuals that in fact are resistant to the antibiotic. So when you apply an antibiotic to a host, one of the things you're going to see is a decline in the sensitive bacteria within that population, and this can lead to antibiotic-associated superinfections. Alternatively, when you apply an antibiotic to just a population, you'll see an increase in the frequency of those individuals that are resistant to the antibiotic. They may not be highly resistant to the antibiotic. If you apply the antibiotic long enough, you'll eliminate all individuals in the population except those that are highly resistant to the antibiotic. Ideally, there will in fact be no individuals that are highly resistant to the antibiotic, and or what individuals are left behind that are resistant will be in such small number that the immune system will be capable of removing them otherwise.